Hello everyone, welcome back to my NicheNet video tutorial. In my video 1, I introduced the NicheNet and also we loaded the NicheNet V2 ligand target prior model. So in this video, I'm going to show you how to perform NicheNet analysis starting from a Seurat object. So first we need to load a Seurat object. I'm going to use the Seurat object that we analyzed in the Seurat video 17 and 20. So in both videos, we integrated healthy non-single-cell RNA sequencing data with fiber optic IPF single-cell RNA sequencing dataset. We named the Seurat object as control IPF combined, so we can load the RDS Seurat object. So you can see in the data windows we have the Seurat object. And let's use the view function to have a look at the metadata. So you can see in the origin ident, we have the samples from healthy lung as the control, and also we have samples from IPF lung. And we named the cell clusters in this Seurat object. In Seurat video 17 and 20, we named the cell clusters as 4 populations: immune cells, mesenchymal cells, endothelial cells, and epithelial cells. Because the actual cellular matrix in IPF lung was largely produced by the fibroblast, so in order to use this dataset for the NicheNet video tutorial, I renamed the cell clusters in more details. So let's use the dim plot function together with the split by argument. We can have a look at the cell clusters in this Seurat object. So let's run the dim plot. We can zoom in. You can see we have cell clusters from the control and also cell clusters from IPF lung. We named the cells as endothelial cells, monocyte, dendritic cells, fibroblast, smooth muscle cells, parasite, NK cells, T cells, epithelial cells, mesothelial cells, cells from the plasma. And we have the same cell clusters from the IPF lung. The dataset contains the cell clusters from healthy donors and also IPF lung. We are going to perform cell-cell communication analysis for the IPF lung. So we can create a Seurat object for the IPF lung. So let's use the split function to split the control and the IPF Seurat object. You can see we created a large list. One is the control, one is the IPF. So we can get the data from the IPF list to create a Seurat object. So let's create a Seurat object for the IPF lung. So we can have a look at the metadata for the Seurat object now. You can see in the origin ident we only have IPF now. And also we can use the dim plot to have a look at the cell clusters. Let's zoom in again. You can see now we only have the cell clusters for the IPF lung. Then we can use this dataset to perform NicheNet cell-cell communication network analysis. So we don't need the list object and also the weighted networks anymore. We can remove it. But we still need the control IPF combined Seurat object because we are going to use this Seurat object to identify differentially expressed genes in the fibroblast in IPF lung. So now we loaded the prior model and also we created a Seurat object so we can perform NicheNet analysis. Because we performed the data integration for healthy and IPF none, at the moment the default assay is integrated, so we can set the default assay as RNA. For NicheNet analysis, we need to define the send and the receiver cell types. So we are interested in the fibroblast cluster, and we want to know what kind of signaling pathways involved in the fibroblast transition into myofibroblast. Because this is a video for demonstration to show you how to use NicheNet and perform cell-cell communication network analysis. So we already know the TGF-beta signaling through TGF-beta receptor 1 and also the IL-1B signaling through the IL-1R1 receptor are important signaling pathways that controls the fibroblast transition into myofibroblast. So we can use the dot plot function to have a look at the TGF-beta and its receptor TGF-beta 1 expression. So let's run the dim plot. Let's zoom in to see the details. You can see TGF-beta 1 is highly expressed in NK cells, dendritic cells, and monocyte, and its receptor TGF-beta receptor 1 is expressed in fibroblast monocyte and dendritic cells. And we can plot IL-1B and its receptor IL-1R1. So let's run the plot and zoom in again. You can see IL-1B is highly expressed in the dendritic cells, monocyte, and its receptor IL-1R1 is highly expressed in the fibroblast and mesothelial cells. So from both TGF-beta 1 and IL-1B signaling pathways, we can see ligand from IPF NK cells, dendritic cells, and monocyte are important ligands that could interact with their receptor on the IPF fibroblast to promote myofibroblast transition. So for this video demonstration to show you signaling pathways that are involved in the myofibroblast transition, so we can set the send cell types as IPF NK cells, dendritic cells, and monocyte. Then the receiver cells will be the IPF fibroblast. So if you want to study the cell communication network between all the cell types, you can just list all the cell types in the send cell types and also all the cell types in the receiver cells. So for today's demonstration, we are going to set the send cell types as NK cells, DC cells, and monocyte. Then receiver cells as IPF fibroblast. So next we need to get all the genes that are expressed both in the send cells and also in the receiver cells. Because we have 3 cell types, NK cells, dendritic cells, and monocyte in the send cells, so we can use the lapply function to get the gene expressed in each cell type, and also we only get the genes that have at least 10% expression in each cell cluster. So you can see we got the data for gene expressed in the send cells, and here we have 3 lists. You can see we have the list for each cell type, and also you can see there are lots of genes. They are expressed in all 3 cell types. So we just need the gene names, so we need to unlist the gene list from raw 3 cell types and make them unique. So let's do this. We can connote some data set. Then we can unlist the genes from all 3 cell types and make the gene unique. So now you can see from the expressed gene center, we have 1704 genes. So next we can get the genes that are expressed in the receiver cells. They are fibroblasts, and also we set the minimum expression as 10% of cells. Because we only have one cell type here, so we don't need to unlist the genes and also make them unique. So let's just get the gene that are expressed in the fibroblast. So you can see from the expressed genes in the receiver, there are 1717 genes in the fibroblast. So to calculate the ligand activities later, we also need to define the background expressed genes. So that's all the genes that are expressed in the fibroblast, and also we can find those gene names in the ligand target matrix dataset. So at the moment, the expressed genes in receiver cells, we have 1717 genes, but we can only calculate the gene activity for the genes that are listed in the ligand target matrix. So we need to get genes that are not only expressed in the fibroblast, but also we have the gene names in the ligand target matrix dataset. So let's find the background expressed genes. So you can see now the background expressed genes is 1629 genes. So this is the number that we are going to use as the background genes. They are all the genes that we can find in the fibroblast and also in the ligand target matrix dataset. So as I mentioned in video 1, if a ligand interacts with a receptor on fibroblast, it will activate this signaling pathway and regulate downstream gene expression. So we need to identify the differentially expressed genes in the fibroblast in IPF-lung. So to do this, we can use the find markers function in Seurat package to compare the IPF fibroblast with the control fibroblast. Because we still have this Seurat object here, contain both control and IPF sequencing information. So let's run the find marks function to identify differentially expressed gene in the IPF fibroblast. So we can set the ident1 as the IPF fibroblast and ident2 as the control fibroblast. And also we set the minimum expression in the fibroblast as 10%. So let's run the find markers function to identify the differentially expressed genes in IPF-lung. So you can see from the DE table, we identified 640 genes that are differentially expressed in the IPF fibroblast. So those differentially expressed genes are the genes that are of interest that we can use for downstream analysis. You can further to refine the genes that are of interest. For example, here we can filter the genes with adjusted p-value smaller than 0.05 and also the log 2FC fold changes bigger than 0.25. So let's get the genes matching this criteria. So now you can see the gene set of interest only has 462 genes. In order to perform downstream analysis to get the weight for the ligand regulate those gene expression in fibroblasts, we also need to make sure that we can find the gene names from the ligand target matrix. So at the moment we have 462 genes of interest. So let's see how many genes we can find from the ligand target matrix from the 462 genes in the gene set of interest. So you can see now we only have 452 genes. So those genes are not only differentially expressed in the IPF fibroblast, and also we can find the gene names in the ligand target matrix dataset. So now we know the gene that expressed in the sender cells and also gene that expressed in the receiver cells. Then we can identify the ligand from the sender cells and the receptor from the receiver cells. So first we can get all the ligands from the ligand receptor network dataset. I showed you in the first video we have 1,226 ligands in this dataset, and also for the receptors we have 1,067 receptors. So now we can use the ligand and the receptor information to find how many ligands expressed in the sender cells. So we can use the intersect function to find the ligand that expressed in the sender cells. So you can see from here we identified 330 ligands that are expressed in the sender cells. And we can do the same for the receptors to see how many receptors that are expressed in the fibroblast. And you can see here we identified 223 receptors that are expressed on the fibroblast. Although we have 330 ligands and 223 receptors between send cells and receiver cells, but the receiver cells not going to express all the receptors for all the ligands that are expressed in the sender cells, and also some receptors that are expressed on the fibroblasts, they may don't have a ligand from the sender cells we defined as dendritic cells, monocyte, and NK cells. So we need to find the total number of ligands that are not only expressed on the sender cells, but also they have receptors expressed on the fibroblast. So let's find the potential ligand. You can see now the potential ligand is 211. And we can have a look at the gene names for the potential ligand. And here is the list for 211 ligand. So we identify the potential ligand, and they have the receptor expressed on the fibroblast. Now we can start to calculate the ligand activities. Use the gene set of interest. That's the 450 genes that are differentially expressed in the IPF fibroblast. So we can use all the genes in the fibroblast as the background genes, and we get the weight from the ligand target matrix dataset for the gene set of interest, and the potential ligand will be the 211 ligand that we identified in the dendritic cells, monocyte, and NK cells. So let's calculate the ligand activities. So you can see we generated a data set called ligand activities. You know we have 211 genes for the ligand. So let's connect the data set to have a look at what's in this data set. You can see the first column are the ligand, the second to the fifth column are different parameters that are analyzed by the ligand activity analysis. So according to the online tutorial, the corrected AUPR is the best parameter to see the ligand activities. The AUPR stands for the area and the precision recall curve. So we can rearrange the corrected AUPR to see which ligand has the strongest activities in IPF fibroblast. So let's rearrange the corrected AUPR. We can arrange it as a descending order. So let's arrange it. We can have a look at the ligand activity again. You can see now the 211 ligand was rearranged according to their ligand activities, and the IL-1B has the highest ligand activity in IPF fibroblast. So we have 211 ligand here. We can get the top 20 ligand as the best upstream ligand, then use the dot plot to see what kind of cell types express the top 20 ligand. So let's run the dot plot for the top 20 ligand genes. Let's zoom in. You can see IL-1B has a high expression in the monocyte and the dendritic cells, and you can see the expression of other ligands in different cell types. So now through the NicheNet analysis, we identified 211 ligand from the send cells, and we also calculated the activities for each ligand that regulate gene expression in IPF fibroblast. So you can do further analysis for a ligand that you are interested in. For example, the IL-1B to identify the target genes in the fibroblast, and also to see the receptor expressions for the ligand. So again, we can use the top 20 genes as an example to perform further analysis. Because we set the top 20 genes as the best upstream ligand, and we know the genes that are differentially expressed in the IPF fibroblast. So we can use the ligand target matrix data to produce a heat map for the target gene expression in fibroblast. So we are going to show the top 20 genes with the heat map, and also we set the maximum target gene as 200. So let's identify the target genes for the top 20 ligand. You can see we generated data called the active ligand target link data frame. Let's have a look at this data set. You can see the first column is the top 20 ligand, then their target genes, and the third column is the weight for the ligand regulates the target expression in IPF fibroblast. So IL-1B is the top target, so we list the gene for the IL-1B first, then next will be IL-1A, then IGF-2. So in this data, we got the ligand target matrix for the top 20 genes. Then we can start to prepare a heat map to show the target genes for the top 20 ligand. So we need to generate a matrix data for the ligand target and also the weight in order to make a heat map plot. You can see we have matrix data here, you can see the column names are the top 20 ligand genes, and the row names are the genes that are regulated by top 20 ligand. So here is the matrix data. If a ligand doesn't regulate this gene expression, the matrix data will be 0. So now we can reward the ligand and also the target genes, and also we get the row names and the column names in order to produce the heat map. So now we can generate a data set that we can plot the heat map. So we can also set all the parameters for the ggplot to plot the heat map as P ligand target network. So let's have a look at the heat map now for the top 20 ligand and their target genes. So let's zoom in. We can make it bigger. You can see now we have the top 20 ligand names on the left hand side, and all the genes that could be upregulated in fibroblast by top 20 ligand. You can see we identified the L1B as the strongest ligand activity, and its target genes show the highest regulatory potentials. So we generated a heat map for the ligand and the target genes, and also we can generate a heat map for the ligand and the receptors. Once again, we are going to see the top 20 ligand interact with their receptors. So we can use the ligand receptor interacting information from the ligand receptor network dataset, and also the receptor that are expressed in the fibroblast to identify the receptors for the top 20 ligands. You can see we named the data as ligand receptor network top, so it is this data set. We can have a look. You can see we identified 45 ligand and receptors interactions. So let's have a look at the data set. You can see the first column are the ligand and the second column are the receptors. You can see here one ligand can interact with different receptors, and possibly one receptor also can interact with a different ligand. So let's make the receptor gene names unique to see how many receptors are there for the top 20 ligand. So we named it the best upstream receptors. You can see the best upstream receptors. We have 37 genes. So now we can use the weighted network prior model to get the weight for each ligand interact with different receptors in our data list. So we have 37 receptors and 20 ligand. So let's get the weight to generate data again. Then we can make a heat map to show the ligand and receptor interactions. So we generated a data set in order to show the heat map. And if a ligand doesn't interact with the receptor, we can fill that gap as 0. So we generate a data set we named as the ligand receptor network top data frame. So let's have a look at this data frame, ligand receptor network top data frame. So that is this one. We know we have 37 receptors. So you can see here is the data frame for the top 20 ligand and the 37 receptors. So we can use this data set to generate a heat map for the ligand receptor interactions. At the moment you can see the receptor names are in the first column, so we can change the class of the data set as a matrix. Then we can run the following code in order to generate a heat map for the ligand-receptor interactions. So now we can visualize the heat map for the ligand-receptor network. Let's run. We can zoom in. You can see now we have the top 20 ligand on the left hand side and the near receptors on the top. And once again you can see strong interaction of L1B through the L1R1 receptor on the fibroblast. So in this video I showed you how to use NicheNet and perform cell-cell communication network analysis. You can see the advantage of using NicheNet. We can prioritize the ligand by calculating their activities in the receiver cells, and the different activity of each ligand was identified by their target gene expression in the receiver cells. So I'm going to stop from here for the demonstration using a Seurat object to perform NicheNet analysis. So I hope my video tutorial can help your data analysis. Please subscribe my channel if you are new to my channel, and also share my videos with your friends. Thank you and see you next time.